Okay, can someone confirm if I'm audible and my screen is visible to everyone?
Yes, ma'am.
Okay. So, welcome everyone. Today is the ninth problem solving session for the course Environmental Chemistry and Microbiology. Till now, we have discussed different topics of environmental chemistry, and from past 2 weeks, we have started the microbiology part. So, we discussed first we had an introduction to microbiology, and then we discussed about cell chemistry and cell biology, so different components of cell and different functionalities. So today we'll talk something more deeper. Now we'll go into the metabolism that occurs inside the cell. Okay, so microbial, mainly we'll focus on the microbial cell metabolism. So microbial metabolism, what all processes are happening, we'll discuss about that. So as you know, course instructors are Professor Anjali Pal and Professor Sudha Goyal from IIT Kharagpur, and I'm Shrija Ghosh, a PMRF from IIT Kanpur. I've prepared certain questions which are related to your assignment questions, and we'll be discussing those now. Okay. So I request all of you to answer the questions when I ask them in the chat box, or you can unmute and answer whatever is comfortable. And then we'll have a discussion if your answers are correct or wrong. At any point, if anybody has any type of doubt, please let me know. I'll try to help you out. Okay. So let's start with the first question. Which of the following terms is used to describe the increase in concentration of a chemical compound in the tissue of a living organism with respect to water? Which of the following terms is used to describe the increase in the concentration of a chemical compound in the tissue of a living organism with respect to water? Options are: option A, live accumulation factor; option B, bioconcentration factor; option C, chemical accumulation factor; and option D, chemical concentration factor. What is the term that is used to describe the increase in concentration of chemical compound in the tissue of a living organism with respect to water? Live accumulation factor, bioconcentration factor, chemical accumulation factor, or chemical concentration factor? What is it called? All of you write your answers in the chat box. Okay, I can see answers coming in the chat box. Four answers are there. All of them are option B. Others also, I request everybody to write their answers. Even if it is the same answer, I want everyone to respond so that I will know if someone has any problem in any of the questions. Okay? Anyone else wants to write? Okay. I have four answers. All of them are option B, bioconcentration factor. That is the correct answer. Option B, bioconcentration factor. So basically, as the name suggests, it is the concentration of any compound in the biological tissue with respect to the concentration of that compound in water. Okay, so bioconcentration factor uh, is defined as the concentration of a compound in the biota. Biota means any living tissue or living organism, divided by the concentration of that compound in water. So, C biota is the contaminant concentration in the tissue of a specific organism in the unit of microgram per kg under equilibrium condition, and seawater is the contaminant concentration in water in microgram per liter. Okay, so why is this factor important? This factor is important because This is how we can determine if a particular contaminant is now accumulating in our tissues more than it should be present. Like there's always a threshold level up to which different living tissues can tolerate that particular contaminant. So you must have heard about metal toxicity. So if there is a metal contaminant, if there's a organic compound with a metal entrapped in it, and if that metal ion is accumulating in our tissues, there's a bioconcentration factor for every contaminant or every compound, every chemical. Uh, there's a bioconcentration factor which is defined as tolerable for a particular tissue. But for certain contaminants, if that level exceeds the threshold level, then we can understand that okay, this particular chemical is now accumulating in our tissue, so it is harmful to us, and now we'll have to do something to get rid of it. Basically, this factor is important so that we can determine whether a contaminant concentration uh, is under the threshold level or it is beyond the threshold level. So we compare that contaminant's concentration uh, with respect to the concentration of that contaminant present in water. So if it is very high in the living tissue as compared to water, obviously it's a problematic situation. Okay, I think this was straightforward. Anybody has any doubt in this? If you have any questions or any doubt regarding any of the questions, please let me know. Please write your doubts in the chat box. Okay. I'll move to the next question. Question number 2. Which of the following is not a xenobiotic compound? Which of the following is not a xenobiotic compound? Options are option A. Option A, dichloro-diphenyl trichloroethane. Option B, trinitrotoluene. Option C, glutathione. And option D, fluorine. Which of the following is not a xenobiotic compound? Dichloro-diphenyl trichloroethane, trinitrotoluene, glutathione, and fluorine. Write your answers in the chat box. All of you quickly write your answers in the chat box. I can see 5 answers already. Rest of you also write your answers and then we'll discuss. There are 15 people in this call right now and only 5 people are answering. What about others? Okay, so the 5 people who have answered option C, glutathione. Glutathione is not a xenobiotic compound. That is the correct answer. So among this list, glutathione is a naturally available compound, whereas all other options are xenobiotics. So xenobiotics are basically the compounds which are not naturally present but synthesized via chemical methods in a laboratory and used for certain different applications. So these are synthetic compounds. These are not naturally produced by any living organism, or yeah, any living organism. Okay, so synthetic organic compounds that are relatively alien or foreign to the environment. Examples: different herbicides, pesticides, PCBs, chlorinated solvents. All of these are synthesized in a lab. Okay, so these are called xenobiotics, and these xenobiotics can be hazardous and toxic because these are not natural substances. So obviously they are foreign to your body, foreign to all the living tissues, and they are toxic to the living organisms. Often toxic to higher and lower organisms, source of pollution problems, short term and long term health effects, depending on the concentration and routes of exposure. So, you know all these pesticides used in the agriculture, and all the chemicals, the chlorinated solvents, all of these things, certain chemicals that are released in the rivers from the industries directly. So all of these are the source of pollution, different uh, pollution related problem and health related problem. So right now, uh, you all are aware of the air quality index, so that is also ultimate effect of pollution, right? Air pollution. Similarly, water pollution, when these chemicals are released in the water. So all the pollutions, and ultimately those pollutions are going to affect your health. So there can be short term as well as long term health effects. So if you breathe polluted air or drink polluted water polluted with these compounds will ultimately end up in your system. And this is where your bioconcentration factor also comes in picture, because then you will have to determine the bioconcentration factor for each pollutant or each contaminant in your like in your cells versus the concentration in water, and that is how you can determine if particular compound or particular pollutant is getting accumulated in your tissue, and then you will have to take certain measures to get rid of it. Xenobiotics in the environment are degraded by physical, chemical, and biological reactions. They can be degraded by certain methods, but they are degrading these compounds is not that simple, and that is again why we are having these kind of bioaccumulation problem and pollution related problems. Okay, because degrade these are synthetic compounds, and degrading these compounds are not very easy. Okay. So the other options that were there, dichlorodiphenyl trichloroethane, which is basically DDT. It's a very commonly used insecticide in agriculture. So uh, whatever agricultural produce you are getting and you are consuming, if they are treated with these kind of insecticides, those insecticides are also getting into your system. That is why it is advised to wash the fruits and vegetables that you are getting from market thoroughly, so that you remove all these kind of insecticide. That is why we are advised to have cooked food or boiled food, so that we at least get rid of certain chemicals. These chemicals are destroyed. Then trinitrotoluene, it's a very commonly used chemical in explosive. These are mostly used by the military uh, systems. And TNT accumulation is also a very big pollution problem, because TNT cannot be degraded very easily, so it remains in the environment and ultimately causes pollution. It's a nitrogen containing compound, so it causes, like, it is very toxic to the living tissue. Then fluorine is also used in dyes and plastics and pesticides and uh, different types of materials. So it is also a xenobiotic compound. Glutathione is a natural compound. It's a peptide found in plants and animals, and it is a powerful antioxidant produced by liver to protect the body against free radicals, peroxides, and heavy metals. Glutathione is actually the compound that is produced by our body to get rid of all these kinds of contaminants and pollutants, all these kind of chemicals. So all the xenobiotic compounds are generally degraded by uh, these glutathione dependent enzymes, and then there are cytochrome P450s. So there are different enzymes that our body is producing to get rid of these contaminants, but still it's not that effective, and then we have certain types of health hazards. Okay, so glutathione eliminates poisons such as drugs and pollutants from our body. Okay, is this clear to everyone? Anybody has any question or any doubt? Please write it in the chat box if you have any question or any doubt. Okay. Moving on to the next question, question number 3. If the octanol water partition log coefficient value of benzopyrene is 6.06, the compound is practically, there are only two options, either it is hydrophilic or hydrophobic. So if the octanol water partition log coefficient of benzopyrene is 6.06, so octanol to water partition log coefficient, so log of the octanol to water partition coefficient, that value is given as 6.06. So you have to tell whether benzopyrene is hydrophilic or hydrophobic. Write your answers in the chat box. I can already see 4 answers. What about others? Only these 4-5 people keep answering, and all other people are just sitting in the class. I don't know why. If you have any doubt, please ask me. Okay. So the answers in the chat box, all of them are option B, hydrophobic. So that is the correct answer, hydrophobic. Uh, so this coefficient, octanol to water partition coefficient, basically tells us if benzopyrene is more soluble in octanol or more soluble in water. Water is a polar solvent. So if it is more soluble in water, it is going to be hydrophilic. And if it is more soluble in octanol, which is a non-polar solvent, it is going to be hydrophobic. Okay. So this is the coefficient, partition coefficient of octanol to water, means the concentration of a particular compound in octanol divided by the concentration of that compound in water. Okay. So if you take the log of this coefficient, this partition coefficient, so the value is given as 6.06. That means the partition coefficient is going to be 10 raised to the power of 6.06. So that means the concentration of benzopyrene and octanol is equal to 10 raised to the power of 6.06 times concentration in water, which is very high. So benzopyrene is obviously more soluble in octanol than water. And here is the relationship between the log partition coefficient in octanol water versus log of bioaccumulation factor. So we talked about the bioaccumulation factor in the beginning, right? So it is directly proportional to this partition coefficient. So as the molecule becomes more hydrophobic, the bioaccumulation factor also increases. And that is logical also, because if you think of it, if the partition coefficient of a particular compound is high, that means it is more hydrophobic. And if it is more hydrophobic, it is not going to be soluble in water, and it cannot be washed off from the system easily. So our body is also in a hydrophilic environment, right? So if something is hydrophobic, it is going to stay there. It cannot be removed from our system easily, because all the excretion process from our body occurs via water, via aqueous system, right? So that's why if a compound is more hydrophobic, it is going to have a higher bioaccumulation factor, and if it is hydrophilic, it is going to have a lower bioaccumulation factor. Is this clear to everyone? If you have any doubt, please let me know. I'll try to help you out. I hope this is clear. I'll move to the next question. Question number 4. How many intermediates are generated in a glycolysis pathway? How many intermediates are generated in the glycolysis pathway? Options are 5, 8, 9 or 10. How many intermediates are generated in a glycolysis pathway? Option A 5, option B 8, option C 9, and option D 10. All of you write the answers. Okay, I'm getting different answers now. I can see some people are answering option D and some are answering option C. 9. Okay. Majority have answered option D, 10. So the correct answer to this question is option D, 10. So there are 10 intermediates in the glycolysis pathway. I don't know how you got 9. So the glycolysis pathway starts from glucose. So here's the whole pathway. So it starts from glucose and it ends at pyruvate, and there are total of 10 steps in this pathway. So from glucose, first step is formation of glucose 6 phosphate. So a phosphorylation reaction occurs. So the enzyme involved is hexokinase. So wherever enzyme called kinase is involved, it is going to add phosphate or it is going to transfer phosphate group. Okay. So here hexokinase transfers phosphate group to glucose to form glucose 6 phosphate. This glucose 6 phosphate then forms fructose 6 phosphate, which is the second intermediate. So from glucose 6 phosphate, it's a isomerization reaction where it becomes fructose 6 phosphate, and the enzyme is phosphoglucose isomerase. From fructose 6 phosphate, we get fructose 1,6 biphosphate or bisphosphate, where one more phosphate is added to this fructose 6 phosphate by another enzyme called phosphofructokinase. Again, a kinase is there. So that means it is involved in a phosphate addition. So one more phosphate is added. So this is the third intermediate. From this, we get two intermediates. Fructose 1,6 bisphosphate uh, is broken down by aldolase into two compounds. One is glyceraldehyde phosphate and second is dihydroxyacetophenone. Okay. So fructose 1,6 biphosphate, it breaks down into two compounds. One is glyceraldehyde 3 phosphate and one is dihydroxyacetone phosphate. These two compounds then dihydroxyacetone acetone dihydroxyacetone phosphate can get isomerized into glyceraldehyde 3 phosphate. So these two compounds remain in equilibrium. All the glyceraldehyde 3 phosphate, they will go and form 1,3 phosphoglycerate. Then from 1,3 phosphoglycerate we get phosphoglycerate to 3 phosphoglycerate. From 3 phosphoglycerate we get 2 phosphoglycerate by another isomerization reaction by the enzyme phosphoglycerate mutase. Then 2 phosphoglycerate gets converted into phosphoenol pyruvate by the enzyme enolase, and then there is a dephosphorylation reaction. From phosphoenol pyruvate we get pyruvate by an enzyme called phosphoenol pyruvate PP kinase or pyruvate kinase. Okay. So there are 10 steps. You can count the number of intermediates. 1 glucose 6 phosphate, then fructose 6 phosphate is 2, fructose 1,6 bisphosphate is 3, GADP is 4, DHAP is 5, 1,3-PG is 6, 3-PG is 7, 2-PG is 8, PEP is 9, and pyruvate is 10. So all of you who are answering option C 9, I hope this is clear to you. Any doubt in this? Any doubt how we got 10 intermediates? Okay. Okay, one person is saying yes, ma'am. So I am assuming others also understood. If anybody has any doubt, I can explain again. I hope you can see 10 steps here. So there are 10 intermediates. There's no doubt in that. If you still have any questions regarding this pathway, I can explain. Okay, we have more questions related to glycolysis, so we'll be discussing more things, more reactions uh, here. So, in detail, still anybody, if anybody has any question, please let me know. Okay. So glycolysis starts from glucose, ends at pyruvate, has 10 different steps. Okay. And I would request all of you to understand the reactions. It would be easier for you to memorize the pathway if you actually understand what is happening at each step rather than just mugging it up. So I'll also explain it in the upcoming slides, but whenever you are studying such pathways, don't try to mug it up. It would be more difficult to remember each step if you just, without understanding, if you just try to mug it up and learn it. Okay. Okay, the next question, question number 5. Which of the following is a crucial cofactor for the reaction in the glycolysis pathway? Which of the following is a crucial cofactor for the reactions in the glycolysis pathway? Options are option A Mg2 plus, option B Cu2 plus, option C S2 minus, and option D N3 minus. All of you write your answers in the chat box. Mg2 plus, Cu2 plus, S2 minus, N3 minus. Okay, I can see many answers. Okay, all of them are option A, so there's no discrepancy here. So we'll see option A, that is Mg2 plus, that is the correct answer. So all of you who are answering option A, that is correct. So Mg2 plus, it is an important cofactor in glycolysis that helps enzymes hexokinase, phosphofructokinase, glyceraldehyde phosphate dehydrogenase, and phosphoglycerate kinase to function and catalyze these reactions. So we just saw the pathway here. So hexokinase, phosphofructokinase, glyceraldehyde phosphate dehydrogenase, this sixth step, and uh, phosphoglycerate kinase. So all these four enzymes, they require Mg2 plus as a cofactor. So that's why Mg2 plus is important cofactor, because 4 of the 10 steps, 10 enzymes, out of 10 enzymes, 4 enzymes require this metal ion. So it is a very important cofactor for this whole pathway. Okay. Anybody has any questions? Moving on to the next question then. If you have any question, write it in the chat box. I'll look into it. Okay. Question number 6. Which of the following reaction yields ADP from ATP in the glycolysis pathway? Which of the following reaction yields ADP from ATP in the glycolysis pathway? Options are option A 1,3-diphosphoglyceric acid to 3-phosphoglyceric acid. Option B fructose 6 phosphate to fructose 1,6 diphosphate. Option C phosphoenol pyruvic acid to pyruvic acid, and option D 3-phosphoglyceric acid to 2-phosphoglyceric acid. Which of the following reaction yields ADP from ATP? Write your answers in the chat box. Okay, I can see many of you have answered option B, fructose 6 phosphate to fructose 1,6 bisphosphate. So you have to see which reaction is yielding ADP from ATP. That means one phosphate is getting so ADP is adenine diphosphate, ATP is adenine triphosphate. So from triphosphate we are getting diphosphate. That means one phosphate is transferred to some substrate. If you see the options, option A, 1,3-diphosphoglyceric acid to 3-phosphoglyceric acid, here 1-phosphate is removed from 1,3-diphosphoglyceric acid to form 3-phosphoglyceric acid, right? So this is a dephosphorylation reaction. So this can't be the correct answer. Second option, fructose 6-phosphate to fructose 1,6-diphosphate. Here fructose 6-phosphate is taking up one more phosphate to become a diphosphate. So this is the correct answer. So all of you are answering option B. This is the correct answer. Uh, so ATP will give one phosphate to fructose 6-phosphate, and then ATP will become ADP and fructose 6-phosphate will become fructose 1,6-diphosphate. Third option, if you see, phosphoenol pyruvate to pyruvate, that is also a dephosphorylation reaction where phosphate is removed from phosphoenol pyruvate to become pyruvate. The fourth option, 3-phosphoglyceric acid to 2-phosphoglyceric acid, here there's no addition or removal of phosphate, only the position of phosphate is changed. So this is also not the correct answer. If you are getting ADP from ATP, that means ATP is giving its phosphate to some substrate. So you have to see which of the options have a phosphorylation reaction occurring. Okay. So glucose, this is the pathway that I had shown from glucose to pyruvate. This is the glycolysis pathway, and you can see there are steps where phosphate is being transferred. So we have two steps where phosphate is being transferred from ATP. One is glucose to glucose 6 phosphate, which is catalyzed by hexokinase. So ATP will give its phosphate to glucose, and ATP will become ADP, and glucose will become glucose 6 phosphate. So this is the first step where ATP is utilized, and the second step where ATP is utilized is fructose 6 phosphate to fructose 1,6 bisphosphate, where again ATP will give its phosphate to fructose 6 phosphate to become fructose 1,6 bisphosphate. Okay. Is this clear? If you have any doubt, please let me know. Okay. Next question, glycolysis starts with dash and ends with dash. Glycolysis starts with dash and ends with dash. Options are option A, one glucose molecule, one pyruvate molecule, and one ATP molecule. Option B, one glucose molecule, one pyruvate molecule, and two ADP molecules. Option C, one glucose molecule, two pyruvate molecules, and two ATP molecules. And option D, two glucose molecules, two pyruvate molecules, and one ATP molecule. Glycolysis starts with dash and ends with dash. What would be the correct answer? I can see most of you have answered option C. One glucose molecule, two pyruvate molecule, and two ATP molecule. That is the correct answer. So I've explained how glycolysis starts from glucose and ends at pyruvate. So glucose is a 6-carbon molecule, right? Glucose has 6 carbon. So from glucose we are getting glucose 6 phosphate, which is also 6-carbon compound. Then fructose 6 phosphate, again a 6-carbon compound. Then from fructose 6 phosphate, fructose 1,6 bisphosphate, which is also 6-carbon compound. Till here we have only phosphate transfers and isomerization reaction. The compound remains, number of carbon in the compound remains same. Okay, so from here glucose to fructose 1,6 bisphosphate, the reaction is balanced. Now from fructose 1,6 bisphosphate we get two products of 3-carbon compounds. So this is a 6-carbon compound, fructose 1,6 bisphosphate, right? From this it breaks down into dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. So both of these compounds have 3 carbon. So 3 carbon goes into dihydroxyacetone phosphate and 3 carbon goes into glyceraldehyde 3 phosphate. This dihydroxyacetone phosphate also gets converted into glyceraldehyde 3 phosphate by a isomerase enzyme. So it again isomerization reaction occurs and we get two molecules of glyceraldehyde 3 phosphate. So effectively from one molecule of fructose 1,6 bis phosphate, we are getting two molecules of glyceraldehyde 3 phosphate. Okay, so that means from one molecule of glucose, we are getting two molecules of glyceraldehyde 3 phosphate. Okay, so now everything from here onwards will be two molecules. Okay, so two molecules of glyceraldehyde 3 phosphate will give us two molecules of 1,3 bisphosphoglycerate. Then we'll have two molecules of 3 phosphoglycerate, two molecules of 2 phosphoglycerate, two molecules of phosphoenol pyruvate, and two molecules of pyruvate. So from one molecule of glucose, we are getting two molecules of pyruvate. Okay, and also if you see as a chemical reaction, also the reaction should always be balanced. So if we have a 6 carbon compound which is glucose and the product is 3 carbon compound which is pyruvate, that means we need two molecules of pyruvate to balance the number of carbons on the reactant side. On the reactant side number of carbon is 6, so on the product side also we need to have 6 carbon. So one molecule of glucose will give two molecules of pyruvate. Okay, so that part is clear. Now the question is about number of ATPs. So I told you there are two ATPs that are utilized in the two phosphorylation steps. So one ATP is utilized in the first step where glucose is converted into glucose 6 phosphate. So ATP gives its phosphate to glucose to become glucose 6 phosphate, and second ATP is utilized in fructose 6 phosphate to fructose 1,6 bisphosphate step, where ATP gives its phosphate to fructose 6 phosphate to become fructose 1,6 bisphosphate. So 2 ATP are utilized here. Now in this step 1,3 bisphosphoglycerate to 3 phosphoglycerate, this is a dephosphorylation step. So dephosphorylation step means one phosphate is lost from 1,3 bisphosphoglycerate and it just only 3 phosphoglycerate is remaining. So first carbon, the phosphate on the first carbon is transferred to ADP to become ATP. Now since there are two molecules of 1,3 bisphosphoglycerate, we'll have two molecules of ADP. So two molecules of 1,3 bisphosphoglycerate means there are two molecules with two phosphates, right? So now two phosphates can be transferred to two molecules of ADP and it can give us two molecules of ATP. Okay, so we get two molecules of ATP from this step itself. And from the last step, phosphoenol pyruvate to pyruvate, this is also a dephosphorylation step. So again a phosphate is lost from this phosphoenol pyruvate. That phosphate is transferred to ADP. Since there are two molecules of PEP, we'll two molecules of ADP can take these two phosphates and give us two molecules of ATP and the final product, that are two molecules of pyruvate. Okay, so we are getting here two molecules of ATP, here also two molecules of ATP. That means we are in total we are getting four molecules of ATP, but we are losing two molecules of ATP in the first step and the third step. So in effectively if we see in total we are getting only two molecules of ATP because we are getting four and losing two. So that means effectively we are only gaining two molecules of ATP. So glycolysis starts with one molecule of glucose and ends with two molecules of pyruvate and two molecules of ATP. Okay, is this clear to everyone? If anyone has any doubt in any of the steps in glycolysis or any of the uh, part in this whole pathway, let me know. Any reaction related doubt or question related doubt? So you understood how just based on the number of carbons, the substrate and the product has to be balanced. So 1 molecule of glucose will give 2 molecules of pyruvate. Okay. Okay. If there's no question, then I'll move to the next question. Question number 8. Under anaerobic conditions, Saccharomyces species can convert pyruvate into the following. Options are lactic acid, option B ethanol and CO2, option C acetic acid and CO2, option D butyric acid and isopropyl alcohol. Under anaerobic conditions, Saccharomyces species, that is yeast, can convert pyruvate into the following. Option A lactic acid, option B ethanol and CO2, option C acetic acid and CO2, option D butyric acid and isopropyl alcohol. Write your answers in the chat box. All of you write your answers in the chat box. I can see 5 answers in the chat box. Others also write your answers. Under anaerobic condition, Saccharomyces species can convert pyruvate into. So the 5 answers are option B. All 5 of you are saying option B. It can convert pyruvate into ethanol and CO2. So that is the correct answer, ethanol and CO2. Option B is the correct answer. So under anaerobic conditions, Saccharomyces can convert pyruvate into ethanol and carbon dioxide by fermentation pathway. So pyruvate actually has 3 fates inside the cell. So this is called the fate of pyruvate. So pyruvate can either enter the aerobic respiration. So we all are breathing oxygen in and releasing carbon dioxide out, right? But what is happening when we are breathing in oxygen and how we are exhaling carbon dioxide? We are basically undergoing aerobic respiration. We are respiring in presence of oxygen. Okay, so aerobic respiration. So in that condition, we are breathing in oxygen because the oxygen acts as the terminal electron acceptor, and uh, it takes up the electrons, and because of that, ultimately we get ATP molecules produced inside the mitochondria, and those ATP are the driving force for our body. So ATP is the energy molecule, energy currency of the cell, and at the expense of ATP only we are able to function. All our organs are able to function. We are able to walk, we are able to talk and do all the work that we are doing. Okay, so that is aerobic respiration. So under aerobic respiration, the pyruvate gets converted into acetyl coenzyme A. That acetyl coenzyme A will enter the citric acid cycle or the TCA cycle, and there we'll get different electron rich electron carrying compounds, NADH, FADH2, and then those will enter the electron transport chain, ultimately producing CO2 and H2O. That CO2 is exhaled out by us, and oxygen will be the terminal electron acceptor. But under anaerobic conditions, there can be 2 fates of pyruvate. One is uh, fermentation pathway, which is carried out by yeast. So in yeast, if we grow the yeast cells in the absence of oxygen, instead of formation of acetyl coenzyme A, the pyruvate will now form ethanol and carbon dioxide. How? by the enzyme called pyruvate decarboxylase. So first pyruvate carboxylase will decarboxylate the pyruvate to form acetaldehyde, and this acetaldehyde will get converted into ethanol. Okay, so this is the process that is used by all the fermentation industry, all the alcohol uh, industries, the industrial production of ethanol as well as alcohol production in beverages. All of the processes are taken up by these yeast cells. So yeast cells are grown under anaerobic condition in absence of oxygen so that it can produce ethanol in larger quantities. Okay, another case that happens inside uh, our body as well is another type of fermentation pathway where uh, our body starts converting pyruvate into lactate by the enzyme lactate dehydrogenase. So, this actually happens when we have vigorous exercise, when we undergo vigorous exercise. If you are running too fast, you feel a pain in your muscles, right? And that pain is due to the formation of this lactate or lactic acid in the muscle cells. So what happens is when we are running too fast or when we are doing some vigorous exercise, in that case, our body is now starving for oxygen. Body cannot supply enough oxygen to those muscles and to those particular tissues where it requires, because when we are running too fast, our body needs energy. For producing energy, our body is now breaking down the glucose quickly to form pyruvate and get more and more ATP. And so much of pyruvate cannot enter the TCA cycle. Like all of the pyruvate cannot enter the TCA cycle by acetyl coenzyme A because there's not enough oxygen to take up the electrons at the end at this point. Okay, so in that case, under the lack of oxygen, the some of the pyruvate, because pyruvate is getting accumulated because glycolysis is happening continuously when we are exercising, because our body thinks that if we are exercising so much, our body has to make ATP, and for making ATP our body is signaling glycolytic pathway. So the accumulated pyruvate then enters this lactate fermentation pathway where the pyruvate, excess pyruvate, gets converted into lactate. That lactate accumulates in the muscle cells, and that's why we feel the pain in the muscles when we are doing heavy exercises. Okay, is this clear to everyone? So these are the fates of pyruvate. So pyruvate can either undergo aerobic respiration where oxygen will take up the electron, pyruvate will get converted into acetyl coenzyme that can enter the citric acid cycle, or pyruvate can get converted into ethanol and CO2 by yeast, or pyruvate can convert into lactate by lactate dehydrogenase that happens in some microorganism and some of the cells in our body as well. Okay, anybody has any questions? Any question, any doubt? Okay, moving on to the next question. Question number 9. What is the efficiency of ATP generation if one molecule of glucose yields 36 ATP? What is the efficiency of ATP generation if one molecule of glucose yields 36 ATP? So delta G is given as negative 686 kilocalorie per mole. So the free energy for the whole reaction is given as 686 kilocalorie per mole, and the delta G for ATP is given as minus 7.3 kilocalorie per mole. So what is the efficiency of ATP generation if one molecule of glucose yields 36 ATP? So from one molecule of glucose we are getting 36 ATP. For the whole reaction, for the whole oxidation of glucose, uh, the energy is 686 kilocalorie per mole. For one ATP molecule, the free energy is minus 7.3 kilocalorie per mole. So what would be the efficiency? How will we calculate? All of you calculate. So for one ATP molecule, energy is given I can see one person has already calculated. Very good. Others also, I request quickly do the calculation. So for one molecule of ATP, the free energy is given as minus 7.3 kilocalorie per mole. So first calculate how much energy is required by 36 ATP. And then you have the total energy given. You will know the total energy for ATP. So just find the percentage of ATP energy from the total energy. So you will get the efficiency. I can see some of you are doing the calculation. Some of you have done the calculation and you have answered option B. 4 of you have answered, all of you have answered option B. Anyone else has done? Anybody else has completed the calculation? Okay, let's see how to do this. Option B, 38%, I think it is the correct answer. So delta G for ATP is given as minus 7.3 kilocalorie per mole. So for 36 ATPs, it will be 36 into 7.3 kilocalorie per mole. Okay. So first you calculate that, and then total energy is given as 686 kilocalorie per mole. So if I have to find the efficiency of ATP generation, I'll have to divide the total energy of ATP by the total energy of the reaction that is given and multiplied by 100 to get the percentage. So this is the free energy of the complete glucose oxidation. So glucose is oxidized into 6 molecules of carbon dioxide and 6 molecules of water. And the delta G for this reaction, the free energy, Gibbs free energy for this reaction is given as minus 686 kilocalorie per mole. Okay. ATP generated from 1 mole of glucose is 36. So 36 ATP will yield 262.8 kilocalorie per mole. Okay, per mole of glucose basically. So 1 mole of glucose is yielding 36 ATP. So this much energy is required by 36 ATP. 262.8 kilocalorie of energy is required by 36 ATP. So what would be the percentage? 262.8 divided by 686 multiplied by 100. So if you solve that, you will get 38.3. So efficiency of ATP generation from 1 mole of glucose by respiration would be 38.3%. Is this clear to everyone? So 4 of you have calculated it correctly. Very good. People who haven't answered, is this clear to you? I hope this is straightforward enough. If anybody has any doubt, please let me know, okay? I'll move to the next question. Question number 10. Electrons released by the citric acid cycle move down to the electron transport chain in which of the following manner? The electrons released by the citric acid cycle moves down to the electron transport chain in which of the following manner? So you have to tell the sequence of the electron transfer in the electron transport chain. Option A, NADH, complex 1, coenzyme Q, complex 3, cytochrome C, complex 4, and then H2O. Option B is complex 1, coenzyme Q, cytochrome C, complex 2, complex 3, complex 4, and then H2O. Option C is NADH, complex 1, complex 3, coenzyme Q, cytochrome C, complex 4, and then H2O. And option D is coenzyme Q, complex 1, NADH, complex 3, cytochrome C, complex 4, and then H2O. What would be the correct answer? Write, everybody write your answers in the chat box. What is the sequence of electron transfer in the electron transport chain? I can see many of you have answered option A. Okay. Anyone else wants to answer? Okay. Option A, that is NADH, then complex 1, then coenzyme Q, complex 3, cytochrome C, complex 4, and H2. That is the correct answer. So, this is how the electron flow occurs in the electron transport chain. So, from the TCA cycle or the citric acid cycle, I told you the acetyl coenzyme A, which forms from pyruvate. So, glycolysis is the first step. Glycolysis occurs where glucose is converted into pyruvate. From pyruvate, it, acetyl coenzyme A is formed. This acetyl coenzyme A enters the TCA cycle or the citric acid cycle, and it forms different electron carriers like NADH and FADH2, which will enter the electron transport chain. So NADH is the electrons from NADH is transferred to complex 1 first. Complex 1 is NADH dehydrogenase, where an electron from NADH will be transferred and NADH will get converted into NAD plus. These electrons are then given to coenzyme Q. Simultaneously, complex 2 will also take up the electrons from FADH2. So, FADH2 has 2 electrons. FADH2 will give the electrons and get converted into FAD plus, and these electrons are also given to coenzyme Q. So, from complex 1 and 2, the electrons are transferred to coenzyme Q. From coenzyme Q, the electron is transferred to complex 3. From complex 3, the electrons are transported to cytochrome C. From cytochrome C, the electrons are then given to complex 4, where from complex 4, the electrons are accepted by the oxygen. I told you oxygen is the terminal electron acceptor. So, here from NADH and FADH2, all the electrons will go to from complex 1 and 2 to coenzyme Q. From coenzyme Q, it will go to complex 3, then cytochrome C, then complex 4. Then from complex 4, these electrons will be accepted by oxygen to form H2O, which is the final electron carrier. And in all these processes, there are protons, you can see H plus ions, which are pumped out of the membrane. So, uh, this is the mitochondrial membrane. So, inside is the mitochondrial matrix, the outside is the intermembrane space. So, there are two membranes present in mitochondria, and this part is the intermembrane space, the space between the two membranes. Okay. So, in this intermembrane space, all these protons are pumped by complex 1, complex 3, complex 4, and the intermembrane space becomes high in proton concentration. Okay. Then comes the complex 5, or the ATP synthase complex. We'll talk about it in the next questions. Okay, upcoming questions. Is this clear to everyone? Anybody has any questions? So you can see all the pathways are interconnected to each other. From glycolysis itself, it begins. From glycolysis, we reach the citric acid cycle or TCA cycle, and from citric acid cycle, we reach the electron transport chain. Okay. Any questions, anybody? Okay, moving on to the next question. Question number 11. The conversion of ATP into ADP and PI is an example of what type of reaction? The conversion of ATP into ADP and PI is an example of what type of reaction? Options are option A, oxidation reduction, option B, amination, option C, phosphorylation, and option D, hydrolysis. Conversion of ATP into ADP and PI is an example of what type of reaction? Oxidation reduction, amination, phosphorylation, and hydrolysis. Write your answers in the chat box. Okay, I can see most of you have answered option D, hydrolysis. That is the correct answer. So ATP is converted into ADP and PI by hydrolysis reaction. So other options, oxidation reduction, amination, and phosphorylation, they are incorrect. Actually, this is a dephosphorylation reaction, and that dephosphorylation occurs by hydrolysis. So what do we mean by hydrolysis? Hydrolysis reaction means H2O or a water molecule will attack a bond. So you can see here the lone pairs on the oxygen of the water molecule, they will attack this phosphate. So ATP has three phosphate group, adenosine triphosphate, right? So you have three phosphates attached. So from these three phosphate, the terminal phosphate will be attacked by the lone pairs on the oxygen of the water molecule. This water molecule will then break this bond between the second and the third phosphate. Terminal phosphate bond will be broken, and one of the hydrogen will attach to this particular H, so you can see, and the OH will get attached to this particular phosphate. So you will get an inorganic phosphate which is written as PI, and you will get one ADP molecule. Okay, so from one ATP molecule triphosphate, you will get adenine diphosphate or adenosine diphosphate plus inorganic phosphate. Okay, so hydrolysis reaction basically means the involvement of water molecule, and water molecule will facilitate the breaking of the bond, and the water molecule will itself get attached to both the constituent compounds that are forming after breaking. So if a molecule is breaking, that means two new molecules are forming. So in those two new molecules, the water molecule itself will break and add. So here you can see the H2O is breaking into H and OH. So this H is uh, attached to the ADP and OH is attached to the inorganic phosphate. Okay, so this is an hydrolysis reaction. This is how ATP hydrolysis occurs. This is clear to everyone. The mechanism is clear to everyone. If anybody has any doubt, please let me know. Okay. Okay, I'll move to the next question. Question number 12. In eukaryotes, while generating ATP molecule from an ADP molecule, the ATP synthase translocate protons from. In eukaryotes, while generating ATP molecule from an ADP molecule, the ATP synthase translocates protons from option A, cytoplasm to mitochondrial matrix, option B, intermembrane space to matrix of mitochondria, option C, matrix to intermembrane space of mitochondria, and option D, cytoplasm to intermembrane space of mitochondria. In eukaryotes, while generating ATP molecule from an ADP molecule, the ATP synthase translocates protons from cytoplasm to mitochondrial matrix, intermembrane space to matrix of mitochondria, matrix to intermembrane space of mitochondria, and cytoplasm to intermembrane space of mitochondria. Write your answers in the chat box, all of you. What would be the correct answer? The translocation of protons from what location the translocation of protons happen by ATP synthase. I have only one answer in the chat box. Anyone else wants to answer? Okay, I have four answers. All of them are option B, intermembrane space to matrix of mitochondria. That is the correct answer. So I told you about the electron transport chain, and all these complexes, complex 1, 3, and 4, they were pumping the protons out from the mitochondrial matrix to intermembrane space. As a result, there is a proton gradient that forms. The proton concentration in the intermembrane space is very high as compared to the mitochondrial matrix. This proton gradient, the high concentration of protons in the intermembrane space and the low concentration of protons in the mitochondrial matrix, drives the proton pump in the ATP synthase. So ATP synthase then pumps these protons from the intermembrane space to the mitochondrial matrix, and this pumping of protons inside actually drives this enzyme ATP synthase to form ATP from ADP. So the formation of ATP is a very energy intensive process, because one inorganic phosphate has to attach to the ADP. We saw the ATP hydrolysis reaction, which was the reverse of this reaction. So one phosphate was breaking down from ATP to form ADP. That is a energy releasing process, and ATP hydrolysis reactions occur wherever energy is required to drive any other reaction. So ATP hydrolysis in turns gives the energy to that particular substrate. Okay, so that is a very energetically favorable reaction. But this reaction ADP to ATP, where inorganic phosphate has to attach to ADP to form ATP, this requires a lot of energy. So from where this molecule will get the energy? So ATP synthase, this enzyme pumps the proton, and that proton pumping acts as a fuel. So imagine that ATP synthase is a car. Then H plus ions are like the petrol for that car or fuel for that car. Okay. So without the proton pump, this enzyme will not work. So ATP synthase actually has a whole mechanism where when once the protons are pumped in, uh, one proton is pumped in and the molecule turns and ADP gets attached, and then another proton gets pumped in and inorganic phosphate gets attached to ADP, and then in the third round ATP is formed and it is released out, and one by one again proton is pumped, one ADP is attached, again proton is pumped, and then phosphate is attached to ADP, and then again ATP is released. So that's how the cycle keeps going on. So as long as these NADH and FADH2 carries the electron from the citric acid cycle and transfers it through these complexes, when electrons are transferred through these complexes, protons are pumped out. Because these protons are pumped out, proton gradient is formed, and that's how these proton gradient uh, drives this proton pump in the ATP synthase. And because of this proton pumping, ATP is formed by this enzyme ATP synthase. Okay, so you can see now how everything is connected. So if glucose is converted into pyruvate by the glycolysis pathway, and pyruvate is converted into acetyl coenzyme A, which will enter the citric acid cycle or the TCA cycle, we will get these NADH and FADH2. And once we get these NADH and FADH2, only then the electron transport chain will function, and the proton pumping outside the mitochondrial matrix will occur, creating the proton gradient, and this proton gradient is now driving the ATP synthase to form ATP. So that is how from glucose to the energy currency, that is ATP, you can see how everything is well connected. Okay, so that is why when we eat something sweet, we suddenly feel very energetic, because our body suddenly has a glucose spike, which ultimately results into excess glycolysis and excess ATP production. So if you are feeling very lethargic, and if you take something some form of sugar in intake, then immediately you will feel a sudden boost of energy, and that energy is actually the result of glycolysis and ATP synthesis. Is this clear to everyone? Anybody has any questions or any doubt? Okay. If you have any questions, please let me know. Okay. Moving on to the next question, question number 13. Which of the following has the lowest E naught values? Which of the following has the lowest E naught value? Options are option A, ubiquinone, option B, cytochrome C, oxidized versus reduced, option C, NAD plus versus NADH, option D, O2 versus H2O. Which of the following has the lowest E naught value? This is the reduction potential. Okay, redox potential. Ubiquinone, cytochrome C, NAD plus NADH, or O2H2O. Shows previous slide. This is the previous slide. This, or you want to see the question?
Yes, ma'am. Thank you, ma'am.
Okay. Yeah. What would be the answer here? Which of the following has the lowest E naught value? All of you write your answers. Okay, I can see most of you have answered option C, NAD plus by NADH. That is the correct answer. So NAD plus NADH has the lowest E naught value. So these are certain biochemical reactions that occur inside our cells, and all of these have a redox potential. So if you see for NAD plus NADH, the redox potential is negative 0.3, whereas the other options, ubiquinone, cytochrome C, and oxygen to H2O, these have a positive redox potential. So the lowest redox potential is of NAD plus and NADH. Okay, is that clear to everyone? Any doubt? Let's move on to the next question. Which of the following statement is not correct about the electron transport chain? Which of the following statements is not correct about the electron transport chain? Options are: option A, coenzyme Q is also known as ubiquinone. Option B, complex 1 is also known as NADH dehydrogenase. Option C, cytochrome C accepts electrons from complex 2. And option D, coenzyme Q accepts electrons from complex 1 and complex 2. Which of the following statement is not correct about the electron transport chain? Coenzyme Q is known as ubiquinone. Complex 1 is known as NADH dehydrogenase. Cytochrome C accepts electrons from complex 2, and coenzyme Q accepts electrons from complex 1 and complex 2. Which of the following is incorrect? Okay, not correct about ETC. Write your answers in the chat box. I can see option C has been answered. Anyone else wants to write the answers in the chat box? Okay. Okay, let's see the correct answer. The correct answer is option C. Cytochrome C accepts electrons from complex 2. I just showed you the electron transport chain, explained all the steps. So, you know, cytochrome C accepts electrons from which complex cytochrome C is accepting electron? Anyone? I just explained the electron transport chain. Yes, complex 3. So, Mopia has answered complex 3. Cytochrome C accepts electrons from complex 3. Complex 1 and 2 gives the electrons to coenzyme Q. So, the fourth statement is correct. Coenzyme Q accepts electrons from complex 1 and 2. Complex 1, I told you, it is called NADH dehydrogenase, and coenzyme Q is also known as ubiquinone. Okay, so coenzyme Q and cytochrome C, these are known as electron carriers in the cell. And complex 1, complex 2, these are enzymes. Complex 1 is NADH dehydrogenase, and complex 2 is called succinate dehydrogenase. Okay, so NADH dehydrogenase, it accepts electrons from NADH and transfers those electrons to coenzyme Q. Succinate dehydrogenase, it accepts electrons from FADH2. So succinate will give the electron to FADH2, and then FADH2 will give the electron to this enzyme succinate dehydrogenase, and then this electron is transferred to coenzyme Q. From coenzyme Q, the pathway, you know, I have already explained this part. Anybody has any questions? This is clear to everyone, right? So I hope you are understanding how these pathways are interconnected, because that is the main part that you have to understand. So everything that is happening so all these things are happening inside your body, so you should actually try to connect with that. Okay, when we eat something sweet, it ultimately gets converted into glucose, then how glucose is giving us energy. So how whatever we are eating, the food is ultimately converted into energy. Whenever I'm talking about energy inside a cell, mostly we are talking about ATP. Okay, so how ATP formation occurs, and how we are breathing in oxygen, and that oxygen is also required for our survival. You can connect all those things. Okay, so whatever is happening inside your body, you can actually think, and you can understand, okay, this is the process that is happening. Okay, Let's move on to the next question. Question number 15. The ATP synthase powers the generation of ATP molecules with the help of. ATP synthase powers the generation of ATP molecules with the help of. Option A, ionic gradient generated by the movement of ions from inner membrane to matrix of the mitochondria during electron transport. Option B, intercellular movement of phosphate ions during the electron transport. Option C, intracellular movement of ATP and ADP molecules. Option D, proton gradient generated by the movement of protons between the matrix and the intermembrane space in the mitochondria during the electron transport. We have already discussed this, so all of you should be able to answer how ATP synthase is generating power, how ATP synthase is getting power to generate ATP. I have already explained this to everyone, so everyone should be able to answer. I still have only 5 people answering out of class of 18 or 19. We just discussed in deep lengths about electron transport chain and ATP synthesis. How is ATP synthase getting its power to generate the ATP molecules? I told you generation of a ATP molecule is an energy intensive process. From where is the energy coming? So 5 of you who have answered, you have answered option D, proton gradient. Obviously we have already discussed this, so there should not be any doubt. Proton gradient generated by the movement of protons between the matrix and the intermembrane space and the mitochondria during electron transport chain. It powers the ATP synthase to generate ATP molecules. Okay, so we have already discussed this, not explaining it again. If anybody has any doubt in this, please ask in the chat box or unmute and ask. I've explained this diagram and the whole process multiple times now. I hope it is clear to everyone, right? Let's move to the next question. Which of the following is a terminal electron acceptor for aerobic reaction? Again, something we have already discussed. Which of the following is the terminal electron acceptor for aerobic reactions? Option A, Fe3, option B, O2, option C, SO4 2 minus, and option D, NO3 minus. Which of the following is terminal electron acceptor for aerobic reactions? We have already discussed this. Terminal electron acceptor for aerobic reaction. Which of the following was accepting electrons? Finally, who accepts the electrons? Fe3, O2, SO4 2 minus, or NO3 minus? Which molecule or which compound accepts the electrons? Which molecule accepts the electrons in the electron transport chain? We saw the electron flow from NADH to different complexes and cytochromes, and ultimately the electron goes to I can see most of you have answered. 4 of you have answered actually. Option B, oxygen, that is the correct answer. So in case of aerobic respiration, I told you oxygen is the terminal electron acceptors. The other 3 options, they are actually the terminal electron acceptors in case of anaerobic reaction. So this is how the different pathways work. So under aerobic respiration, we have oxygen as the terminal electron acceptor, and phosphorylation occurs for ATP generation in 2 processes. One is substrate level phosphorylation. Substrate level phosphorylation is the process where ATP is generated by addition of phosphate via some substrate. So Remember the two ATP molecules that were formed in glycolysis pathway? So that is substrate level phosphorylation, because in case of let's say from 1,3 bisphosphoglycerate to 3-phosphoglycerate, one ATP was formed, right? And again phosphoenol pyruvate to pyruvate, one ATP was formed. So in those reactions, phosphoenol pyruvate is acting as the substrate that is giving the phosphate to ADP, and ATP is formed. Okay, so those are substrate level phosphorylation. And oxidative phosphorylation is the process, that is the electron transport chain, where oxidation reaction is happening, electron transport is happening. Electron transport is basically a part of oxidation reaction, right? So we define oxidation and reduction by the transfer of electrons. So if electron is removed, then we have a oxidation. If electrons are acquired, then we have a reduction, right? So oxidative pathway, under oxidative pathway we get almost 36 ATP, and in the substrate level phosphate for phosphorylation we were getting 2 ATP molecules. So total 38 ATP molecules are generated approximately. Then under anaerobic reaction, now instead of oxygen, terminal electron acceptors will be Fe3 or manganese or SO4 2 minus or NO3 minus, so sulfate, nitrate, all these molecules will accept the electrons, but the process will be more or less same. So instead of oxygen, these molecules will act now, and the number of ATP generated is less than the aerobic respiration, but it is somewhere between 2 and 38. Okay, and the last pathway is fermentation, which we have discussed. So remember fermentation in case of yeast, where pyruvate was converted into ethanol. So pyruvate being an organic compound, the terminal electron acceptor is ultimately organic compound, and there ATP production was only dependent on the glycolysis pathway. Substrate level phosphorylation is happening, and only two molecules of ATP. So fermentation is not a very energy uh, generating molecule. Okay, so in when fermentation is occurring, not a lot of energy is generated inside the cell, whereas aerobic respiration gives the highest amount of energy, then comes the anaerobic respiration, and ultimately the fermentation. Okay, fermentation also occurs anaerobically, but in that case the terminal electron acceptor becomes a organic compound, and only substrate level phosphorylation is happening, no oxidative phosphorylation is happening. Okay, anybody has any questions or any doubt in this? This is clear to everyone, I hope. Okay, let's move to the next question then. Oxidation of N-octane to acetyl coenzyme A is initially catalyzed by which of the following enzyme? Oxidation of N-octane to acetyl coenzyme A is initially catalyzed by which of the following enzyme? Options are option A phosphorylase, option B monooxygenase, option C lipase, and option D catalase. Oxidation of N-octane to acetyl coenzyme A is initially catalyzed by which of the following enzyme? Phosphorylase, monooxygenase, lipase, and catalase. Oxidation of N-octane to acetyl coenzyme A. Which enzyme catalyzes the oxidation of N-octane to acetyl coenzyme A? Write your answers in the chat box. Nobody has an idea? Try to guess the answer. Okay, I have 2, 3. Okay, I'm getting answers now. 3 people have answered option B, that is monooxygenase. Anyone else? Okay. So monooxygenase is the correct answer. So monooxygenase, by the name itself you can understand it, this enzyme can add one extra oxygen to the molecule, so it can actually add OH. So monooxygenase generally add OH to the molecule. So N octane, which is an alkane, it will become N octanol alcohol. Then from alcohol we'll get aldehyde and then carboxylic acid. Okay. And this carboxylic acid, from an alkane, if we get a carboxylic acid, it is a fatty acid, and then it undergoes fatty acid oxidation, that is the beta oxidation, which is a fatty acid oxidation with fatty acid metabolism pathway. Okay. So N octane is catalyzed or is oxidized by monooxygenase enzyme. This is clear. Next question is, steps of anaerobic degradation of alkene follows which of the following steps? Uh, Alkene goes to aldehyde to acetate. Option B, alkene goes to alcohol to acetic acid. Option C, alkene goes to alcohol goes to aldehyde to carboxylic acid. And option D is alkene to aldehyde to alcohol to carbon dioxide. Just showed you the pathway, so it is similar. Quickly write the answers in the chat box. We just discussed the N-octane metabolism, right? So it is similar to that. What would be the first product after alkene? Write your answers in the chat box. We just discussed this, right? The previous question was about this only. What do we get after alkene? I have only one answer in the chat box. When I ask you if you have any doubt or not, nobody asks me any question. But then the next moment when I'm asking a question, why are you not answering? If you understood the previous question, you should be able to answer this question. Only one answer is there in the chat box. Mobini has answered option C, that is alkenes to alcohol to aldehyde to carboxylic acid. That is the correct answer. I just showed you the N-octane to acetyl coenzyme pathway. So from alkane or alkene, it gets converted into alcohol first and then aldehyde, then carboxylic acid. Once you get the carboxylic acid, it becomes the fatty acid, and then fatty acid metabolism by beta oxidation occurs to form the acetyl coenzyme. Is this clear to everyone now? Is there any doubt in this? Any questions regarding this? If there are no doubts, then why were you not answering? This is clear, right? Anybody wants to ask anything regarding this? Okay, otherwise we can move ahead. Next question is, synthetic organic chemicals found in the soil that do not occur naturally are called. Synthetic organic chemicals that are found in the soil that do not occur naturally are called. Options are antibiotic, xenobiotic, chemobiotic, all of the above. This is also something we have discussed in one of the previous questions. Synthetic organic chemicals found in the soil that do not occur naturally. What are those called? Antibiotic, xenobiotic, chemobiotic, and all of the above. What would be the correct answer? Again, there are only two answers in the chat box. What happened to other people? We have already discussed this, right? Okay, five of you have said option B, xenobiotic. That is the correct answer. So I told you xenobiotics are the synthetic organic compounds which are not available naturally. So these are the compounds that are synthesized chemically in a lab and then used for different applications, but they tend to remain in the nature because there's no way of degrading them, because nature evolves ways to degrade things which are present in nature. But if you introduce a new element or new compound which the nature hasn't seen earlier, obviously it will take time for evolution to work its way and degrade those particular compounds. So all the synthetic organic chemicals, these are mostly pollutants because there's no natural way to degrade them quickly. Okay, moving on to the next question. Match the following enzymes with its substrate. We have enzymes on the left hand side: hexokinase, pyruvate kinase, enolase, and aldolase. And we have substrates on the right hand side: fructose 1,6 bisphosphate, 2 phosphoglycerate, phosphoenol pyruvate, and glucose. So match the substrate and the enzyme. Okay, so we have discussed glycolysis in great lengths. So tell me the answer. Match carefully. You have to match the substrate, okay, not the product. Substrate and the enzyme. I can see three answers in the chat box. Option D. Okay. Okay, I can see all of you answering option D. Let's see. So hexokinase is the first enzyme in the pathway. So hexokinase, substrate for hexokinase is glucose. So 1 is 4. Okay, 1 is 4. We have two options. Then pyruvate kinase. Pyruvate kinase is the last enzyme in the pathway, and the substrate for pyruvate kinase is phosphoenol pyruvate. Phosphoenol pyruvate gets converted into pyruvate by the enzyme pyruvate kinase. Pyruvate kinase takes up the phosphate from this phosphoenol pyruvate and gives it to ADP to form ATP, and we get the product pyruvate. Then enolase, substrate for enolase is 2 phosphoglycerate. So 2 will be 3 and 3 will be 2. Okay, so option D is the correct answer. Enolase, 2 phosphoglycerate is converted into phosphoenol pyruvate. And then aldolase, the substrate is fructose 1,6 bisphosphate, where it is broken down into two molecules: dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. Okay, so option D is the correct answer. So all of you have answered. Very good. So again, we have discussed this. I have just added it for your reference. So hexokinase, or sometimes it is called glucokinase. So hexokinase is a general class of enzyme which can phosphorylate any hexose sugar, uh, not just glucose. So glucose is a 6 carbon sugar. There are other 6 carbon sugars. So hexokinase can actually phosphorylate those sugars as well. Glucokinase is a more specific name for the hexokinase that catalyzes the phosphorylation of glucose. Okay, so hexokinase substrate is glucose. Then we had, what did we have? Pyruvate kinase, the last enzyme. The substrate is phosphoenol pyruvate, product is pyruvate. Then We had enolase. The substrate is 2-phosphoglycerate, the product is phosphoenol pyruvate, and then we had aldolase. The substrate is fructose 1,6 bisphosphate, and there are two products, dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. I hope this is clear. We have discussed glycolysis also multiple times today, so I hope this is clear to everybody. Question number 15. Option D is the correct answer for this. Do you have any doubt in this, Trisha? Okay. Okay. Moving on to the next question. This is the last question. Okay, no problem, Trisha. Okay, let's discuss this last question of this session. How many reactions that occur in the TCA cycle transfer electrons from a substrate to an electron accepting enzyme? How many reactions that occur in the TCA cycle transfer electrons from a substrate to an electron accepting enzyme? Options are 1, 2, 3, and 4. I have two answers in the chat box. One says option A and another says option D, 1 and 4. How many reactions are there in the TCA cycle that transfer electrons from a substrate to an electron accepting enzyme? All of you write your answers in the chat box. I can see most of you have answered option D, 4. That is the correct answer. So TCA cycle has 4 reactions. So I told you from glycolysis we get pyruvate. Pyruvate is converted into acetyl coenzyme A that enters this TCA cycle or citric acid cycle. So this acetyl coenzyme A enters the citric acid cycle, and then there are 4 reactions that give NADH and FADH2, which ultimately goes into the electron transport chain. If you remember the electron transport chain, NADH and FADH2 were the electron carriers, which were transferring the electron to the complexes and further carried by the coenzyme Q and cytochrome C, and ultimately accepted by oxygen, right? So these NADH and FADH2 has to form from certain reactions. So you see acetyl coenzyme A enters the citric acid cycle, and by the enzyme citrate synthase it gets attached to the oxaloacetate. So oxaloacetate is a 4 carbon compound, see, and acetyl coenzyme A has 2 carbons. So total we get a 6 carbon compound citrate. Okay, so from citrate synthase, acetyl coenzyme A joins with oxaloacetate to form citrate. Then citrate gets converted into aconitate by the enzyme aconitase, and isocitrate. So citrate to isocitrate is catalyzed by aconitase. From isocitrate we get alpha ketoglutarate. So this is a 6 carbon compound isocitrate. From this the first decarboxylation reaction occurs. So it's an oxidative decarboxylation process, uh, where this CO2 is removed, and we get this terminal CO2 is removed, and we get a 5 carbon compound called alpha ketoglutarate, and this reaction yields the first NADH. Okay, so isocitrate dehydrogenase yields the first NADH. Okay, then the second NADH is achieved from the from the formation of succinyl coenzyme A from alpha ketoglutarate by the enzyme alpha ketoglutarate dehydrogenase complex. So this is again a decarboxylation reaction. Now this 5 carbon compound becomes 4 carbon compound. You can see 1 2 3 and 4. So succinyl coenzyme A has 4 carbons. One carbon is removed as carbon dioxide, and NADH is formed. Okay, so second NADH is formed by this enzyme alpha ketoglutarate dehydrogenase. Then from succinyl coenzyme A we have succinate. From succinate we have fumarate. So succinyl coenzyme A synthetase forms the succinate. Then succinate is converted into fumarate by enzyme succinate dehydrogenase. Do you remember which what was succinate dehydrogenase? I have mentioned this before. Anybody can tell me what is succinate dehydrogenase? I have mentioned the name of succinate dehydrogenase before. Do you remember in what context? Anyone? It is one of the complex in the ETC. Which complex is succinate dehydrogenase? Yes, it is the complex 2. Very good, Mopia. So complex 2 is succinate dehydrogenase. So succinate dehydrogenase, it converts succinate to fumarate, and in that process forms FADH2. The electrons from FADH2 are then transferred to coenzyme Q by this enzyme succinate dehydrogenase. Okay, so succinate dehydrogenase will transfer the electron from FADH2 to coenzyme Q. Okay, so this is the complex 2, which is a part of the citric acid cycle. Okay, so now the third electron carrying moiety is released. From fumarate we have the enzyme fumarase that converts fumarate to malate. Now we have come to the 4 carbon cycle. Okay, so malate to oxaloacetate, we have the enzyme malate dehydrogenase. Again this enzyme will give the third NADH, and in total we have 3 NADH and 1 FADH2. So 4 electron carrying molecules that will ultimately enter the electron transport chain. Okay, and then again oxaloacetate will bind to one more acetyl coenzyme A to form citrate, and the cycle will keep on going, generating more and more NADH and FADH2. Okay, is this pathway clear to everyone? If you have any doubt, please let me know. So you see all the enzymes that has dehydrogenase in their name, mostly all such enzymes are involved with NADH. Okay, so NADH dependent enzymes are generally named as dehydrogenase. Okay, because there's a dehydrogenation reaction happening here. Okay, is this clear to everyone? The pathway citric acid cycle is clear to everyone? So now you understand how from glycolysis we get pyruvate, glucose to pyruvate, then pyruvate to acetyl coenzyme A, then acetyl coenzyme A enters the citric acid cycle to generate NADH and FADH2. This NADH and FADH2 enters the electron transport chain, where ultimately the electrons are accepted by oxygen. Proton gradient is formed. That proton gradient drives the ATP synthase to form ATP. So all of this process is ultimately happening to for the formation of ATP. Clear everybody? Anybody, if anybody has any doubt, please let me know. This was the last question of the session. So, if you have any questions or any doubt in any of the topics of this week, please ask now. If there's no doubt, then we can end the session. Anybody wants to ask anything? Okay, then. Thank you everyone for joining. I'll close the call now. I hope everything was clear from today's session. Okay. I'll see you next Tuesday. Bye bye.
Thank you, ma'am.
Yeah, thank you.